start again. So this is GATE 2022 Life Sciences Microbiology question number 72. Which of the following methods are used for the identification of microorganisms? Option A is nucleic acid hybridization. Option B is southern blotting. Option C is 16S rRNA, ribosomal RNA sequencing. And option D is percentage GC content. Okay, so this is the NPTEL online course reference for this question. So we'll briefly go over all these topics. So what is nucleic acid hybridization? Okay, nucleic acid hybridization. So hybridization is a technique where we have a double-stranded DNA. Okay, we have a double-stranded DNA. It is denatured through heating. Okay, it is denatured to heating. So we preferably heat it at 90 degrees Celsius for 5 minutes. Okay, we preferably heat it at 90 degrees Celsius for 5 minutes on a heating bath. So we have a double-stranded DNA here, and when we heat it at 90 degrees Celsius, we'll get two single-stranded DNA. Okay, this is one template and this is another template, single-stranded DNA template. This. Okay, so we have single-stranded DNA, and this can be again renatured. So if we keep this mixture at a water bath at room temperature, in water bath at room temperature, it will slowly anneal. Okay, this process is known as annealing. This process is known as annealing. It will slowly anneal and it will form a duplex DNA again. Okay, and we can do this number of times. Okay, and it will not damage the DNA or it will not break any DNA. Okay, this denaturing and renaturation process is frequently done in molecular biology labs. Okay, then if we need to hybridize this DNA, what we will do? So we have a single-stranded DNA. If we need to hybridize this DNA with the RNA, okay, and make a DNA RNA hybrid, okay, like this here, then what we'll do, we'll add this RNA mixture. Okay, so if we add this RNA mixtures, there are three possibilities that can happen, right? There are two possibilities that can happen. So we have we can get either duplex DNA again, double-stranded DNA. When we re-anneal it, it will again form a hydrogen bonding with its complementary DNA, right? It will form a hydrogen bonding with its complementary DNA, or it can form a DNA RNA hybrid like shown here. It can form this DNA RNA hybrid here. Okay, so in order to get this DNA hybrid in higher amounts, what we can do, we can add at least 5 times concentration of this single-stranded RNA, 5 times the concentration of. So if we are taking this 10 nanomoles, we can add either 100 or 50 nanomoles. Okay, 50 nanomoles of this DNA. Right, I'm using here 10 times and 5 times that of 5 times that of this DNA concentration. Okay, we have to add this much amount of DNA, so in order to get a DNA RNA hybrid. Okay, and why do we do this technique? This technique is mainly done to label DNA. Okay, and the labels could be of 3 types. The labeling of DNA, we could have a fluorescent label, we could have a fluorescent labeling, we could have a radio labeling, radioactive labels, and we could have a enzyme labeled. This enzyme could be HRP, horseradish peroxide. Okay, we could have a horseradish peroxide template, and if it is a fluorescent label, we just need to fluoresce it. Okay, if it is fluorescent label, generally we use a FAM labeled DNA. Okay, FAM labeled DNA, please go and check what is this FAM labeled DNA. Radioactive isotopes we can use. These radioactive isotopes are generally, the phosphorus is radioactive. It is either P33. Okay, the phosphorus of the DNA, the phosphate group, the phosphorus is radioactive, and then we can use autoradiography technique, right? Autoradiography technique. Autoradiography means the DNA will have a auto-radiation in presence of autoradiography, right? It will radiate few atoms. This process is known as autoradiography in presence of radiation, right? And then there is a enzyme. So instead of FAM, there could be a enzyme. Okay, it could be bound to an enzyme, and to this, when we add a substrate, a chromogenic substrate, we could get a color. Substrate bound to a color, it will give a color. Okay, so these are the different techniques. So nucleic acid hybridization has many applications. Okay, one of the so if we have a DNA, so if we wanted to identify microorganisms, okay, so if you identify a microorganism specific, we can have this DNA, which is this DNA sequence, which is highly specific to that microorganism and which could be FAM labeled or radioactive labeled or enzyme linked. Okay, and then when it is hybridized to the unknown sample, so if we in a tube, if we have an unknown sample of DNA, and if we do this nucleic acid hybridization with our target probe, okay, this is a probe is something which is FAM labeled, okay, which could be used to check. Probe is nothing which could be used as a final product to check the target sequence. Okay, so if it is matching the target sequence, it will either fluoresce or give a autoradiography signal. Okay, and this will help us to identify microorganism. Further, nucleic acid hybridization has many other importance, like Southern blotting. It is also a part of nucleic acid hybridization. I'll explain you what is Southern blotting, which is Southern blotting. Then PCR is also an example of nucleic acid hybridization. Then we have real-time PCR also. These are the other applications for hybridization, nucleic acid hybridization. Northern blotting and southern blotting technique. So northern blotting is nothing to it is used to detect RNA, whereas southern blotting is used to detect DNA. So the scientist, his surname was Southern. So that's he developed this technique of southern blotting to hybridize DNA. Okay, and other applications of this are, I'll just change this slide. Yeah, so other applications of this are fluorescence in situ hybridization, right? We can hybridize this DNA inside of the cell. So these are the chromosome. Okay, this blue color are chromosome, and in these chromosome there are few regions which are hybridized with our FAM labeled, which are fluorescing red. Okay, they are giving red fluorescence here, and the chromosomes which are blue are fluorescing with DAPI. Okay, DAPI is staining this whole cell, and the part of gene which is inside this cell is showing red fluorescence. So this is also example of fluorescence in situ hybridization. Yeah, so northern blotting is RNA. So both northern and southern blotting are applications of nucleic acid hybridization. And then there is one important technique, DNA microarray. Okay, DNA microarray is a high throughput technique. What do you mean by high throughput? Like you can analyze at a time thousands and thousands of sample. Okay, so suppose this well contains a sample from one strain of microorganisms. Like this, we have n number of strains of these microorganisms, and at a single given time we analyze multiple thousands of samples at a time. Okay, so this is a microarray chip. Okay, on this we just spot our samples. Different samples, and then we do this nucleic acid hybridization. Okay, so DNA microarray is also very high throughput. So this high throughput term means that we can analyze n number of samples at a given time. Then there is Southern blotting. So in Southern blotting, we have a DNA, chromosomal DNA. So here the example given is suspect 1, or we can have some microorganism, okay, some X microorganism. We want to identify if this chromosome belongs to that microorganism, right? So what we'll do a restriction digestion, doing a DNA we do a digestion, and we'll divide this huge DNA, because this size is very huge, around 3 to 4 kb, right? Any bacterial DNA. And we cannot run this in the gel. This is very huge chromosomal thick DNA. So we'll cleave it with restriction digestion. It will give us a specific different length of fragments, okay? We can cleave it with either 2 or 3 restriction digestion enzyme, and it will give us a signature pattern, okay? And then here we can use, we will have different suspects of DNA, and we know their pattern, how the pattern looks like. So here we have a suspect microorganisms, that this could be either E. coli or Klebsiella or Salmonella typhi, okay? And we could run, so similarly we could run our control chromosomal DNA, we could digest it with same restriction digestion, RE digestion, okay? And then we run them along with our test chromosomal DNA, okay? Then these are denatured and then transferred onto a nylon membrane. This is a nylon membrane, okay? It is transferred onto a nylon membrane. Then we can either radio label by using a DNA probe, or we can use a fluorescently label, or it could be either enzyme linked, okay? Very simple. And then this is, finally we can expose it to X-rays, different radiation, to emit autoradiography, and this is the autoradiographic signature, right? This is how it looks. Or we could use a FAM labeled, we could see the fluorescence under the scanner, which scans the fluorescently activated probes, okay? This is also an example of nucleic acid. This uses the technique of nucleic acid hybridization. Northern blotting is, we, instead of chromosomal DNA, we'll have RNA. We'll have a RNA. RNA also we have to digest it with restriction enzymes, or we can cleave it with different RNases. We can cleave it with different RNases and then separate it on agarose gel electrophoresis, transfer it to the nylon membrane, do the radioactive labeling or fluorescent labeling, and then visualization. The next is 16S ribosomal RNA sequencing, okay? So in 16S ribosomal RNA sequencing, what do we have? We have a template of unknown sequence, right? We have this template of unknown sequence. Suppose we have a template of some X microorganism which we want to identify, right? Then we have a primer here, okay? We have a primer. Primer will be specific to that ribosomal RNA, okay? This is ribosomal RNA, 16S ribosomal RNA. So we'll have a specific primer. Why 16S rRNA? Because this ribosomal RNA is conserved among many species. It is very much conserved among many species of microorganisms. So we'll use that specific primers. It will polymerize DNA. After adding NTPs, we'll denature this. Okay, we'll denature this, and this will denature this, and then we'll analyze the fragment length. Okay, we'll analyze the fragment length, and then we can check. Then we'll have the sequencing result. Okay, we'll have the sequencing result, and this sequencing result will give a specific signature. This is very important. It will give us a specific signature. So the ribosomal DNA of E. coli will have a specific signature. The ribosomal DNA of Klebsiella will have a specific signature, okay, and that specific signature is very properly curated by any software, or so some computer generated software, or by using simulation it is properly curated, which helps us recognizing. Okay, this all database is stored in the NCBS database, all the database, and these simulations and software utilize the data from these databases, and then they by looking at the signatures they can confirm, okay, this X organism is actually E. coli, or this X organism is Klebsiella. Okay, this is also one of the application related to nucleic acid hybridization. It utilizes the primer extension method, okay, somewhat like PCR. Coming back to the question, the percent GC content. Okay, so this percent GC content is more of related to the whole genome, and it is mainly seen in it is mainly same in the prokaryotes. Okay, this percent GC content, because this is prokaryotes. Why? So this GC content is conserved in the prokaryotes. Okay, so in case of E. coli, Klebsiella, the percent GC content will be somewhat same. It is conserved in prokaryotes, but the percent GC content of prokaryotes and a human cell will vary, right? Due to complexity, due to DNA complexity, the percent GC content may vary. So it's not an ideal tool. In fact, you cannot determine to identify the any microorganisms. Okay, so which of the methods are used for the identification? It's mainly A, B and C. Okay, so the correct answer is A, B and C, whereas percent GC content just tells us about how complex the DNA content is, okay, in any cell. Okay, it tells us about complexity. It tells us about complexity of genome.